stable compound to release. Hydroxide radicals can form. These guys, since they're free radicals, are very reactive. And they will go on and react with stuff, right? So they will generate what we can call oxidative damage. Question?
I'm just looking at your arrows when you go from H2O2 to OH radical.
Dot OH, yes.
You're adding an H in, is that backwards? H2O and H plus? Am I being an idiot?
We are taking out an H and taking out an O. So I think it's going from H2O2 to HO.
So the water's coming out.
Water's coming out, one oxygen's coming out, two hydrogens are coming out, one proton's going in, and an electron's going in. I think it balances out. It's not an important reaction, it's not enzyme catalyzed, but it's just kind of what happens. I mean, it's important to know that hydroxide radical gets generated from hydrogen peroxide. So cells do have enzymes called catalases. And this is a class of enzymes. I don't think we'll need it again. But this is an enzyme that will catalyze the decomposition of peroxide into water and molecular oxygen. And I know that the stoichiometry of that does not balance, but there's a complicated mechanism involved that we're not really going to go into. Okay, but just to know that that's another way that H2O2 can get detoxified. So third way, so we haven't, I'm doing this because I'm explaining NADPH, and we haven't done anything with NADPH yet, but here it comes. So there's an enzyme here called E3. So down here somewhere we'll say E3 is glutathione peroxidase. Okay. So what this enzyme does is it detoxes hydrogen peroxide. And the way it detoxes hydrogen peroxide is it makes use of a pretty ubiquitous cellular redox buffer cofactor. And that's called glutathione. So 2 molecules of reduced glutathione generate 2 molecules of oxidized glutathione. And I will tell you what this G is. But you can see that the reaction, the redox aspect of this reaction is that we have sulfhydryls getting oxidized to an SS bond. So a disulfide bond. Okay. So here, this is clearly the reduced form, this is the oxidized form. Okay. And what glutathione looks like is it's a tripeptide, and it's linked, one of the linkages is through the gamma carboxylate of glutamate. This is not a tripeptide that's made in a coded way on the ribosome, which is only 3 amino acids long, and it's got one linkage that is not a peptide linkage. It's this weird thing through the end of the glue. Okay. This is the end of the glue here. This will be the alpha carbon on glutamate. This will be the gamma carbon on glutamate. And then here would be a cysteine. And here's a glycine. So glutathione is gamma glu cis gly, a tripeptide of that sequence. And we say gamma glu just to indicate that when you get to the next amino acid, rather than NH going to the alpha carbon derived carboxylate on glutamate, it goes to the side chain. Right? So this is reduced glutathione, and then the oxidized form would look like this. So there's 2 molecules of it. Okay, so 2 of these go to this. Okay, so this is it's a buffer in the cell because it has titratable groups that pick up and release protons and help to buffer the cell's pH. Right? But it's also a redox compound because it can oxidize and reduce through its cysteine. So this is a pretty ubiquitous compound inside of cells. You really should be well aware of it. It's often left out in discussions of some parts of biochemistry, but it's very important. It ties into a lot of other kinds of reactions because it's always there, and it's there in substantial amounts. So it's buffering, and it's doing, if there's electron transfer reactions to get handled, and maybe there aren't other specific proteins to do it, maybe this buffer system will come into play. Yes, sir?
This is the same E3 that is involved in ubiquitination.
No.
Because you keep saying ubiquitous.
Oh, I'm sorry. Yeah, I didn't mean to imply there was a link to ubiquitin. There's not. No, ubiquitin is a system that uses E1, E2, E3. I mean, just as part of its nomenclature. It's a system for tagging proteins so that they can go die in the cells. This has nothing to do with that. I'm just writing E1, E2, E3 as a way to keep track of the enzymes. All right, so that's what glutathione looks like. Okay. And to get back to this, the question is, what happens? So the peroxide has been detoxed to water, right? It's been oxidized. It's been reduced. The glutathione has been oxidized. All right, so now we have oxidized glutathione. And we have another enzyme, E2. And here's the NADPH. And E2 is glutathione reductase. Okay, so there's your role for NADPH. And then we could have, here we would have E1, for example, where E1 is glucose 6-phosphate dehydrogenase from the pentose phosphate pathway. There could be other E1s. Any enzyme that's generating an NADPH could be an E1 in this reaction. But since we're talking about pentose phosphate pathway, we can say glucose 6-phosphate goes to 6-phosphoglucono-delta-lactone. Don't worry about what that's actually called. We'll see it on the slide. It's an intermediate in that process. Okay. So this is kind of an introduction to what's going on with oxygen detoxification. It's quite important in aerobic cells to be aware that the cell has mechanisms to take care of this kind of stuff. Okay. Any questions about this? Let's see, there's one other point here. This peroxidase here this is another general class of enzymes. And they work on peroxides like hydrogen peroxide, but they also work on other kinds of peroxides. And this has important roles in some aspects of fatty acid metabolism that we're going to touch on in a couple of weeks. But if you just imagine there's 2 R groups on a peroxide, it could be hydrogens, like hydrogen peroxide. And there's some electron donor, right, to reduce them. Right? And the electron donor varies with the enzyme, or with the, which peroxidase we're talking about. So of course, anytime we write a reaction, we just write E minuses. What we mean is that there's some electron donor, right? Not that there's probably, almost certainly not there's free electrons jumping on, right? Except for those non-specific reactions. In that case, there might be a free radical kind of thing happening. But in any sort of controlled enzymatic reaction, we're going to be having a redox cofactor, right? So what would be an example